Hello, everyone, and welcome to my presentation over the systematic variance of the number of dip ligands and their effect on cytotoxicity and lipophilicity. For my project, I was tasked with determining how the ligand 4,7-diphenyl-1,10-phenanthroline, otherwise known as the dip ligand, affects cellular uptake and cytotoxicity. And my objectives of this project were to synthesize 4 ruthenium polypyridine complexes such as Rutris dip, Ru dip 2 bipi, Ru bipi 2 dip, and Ru tris bipi, with the bipi ligand being 2,2 bipyridine. And my next objective was to conduct LogP assays on the RPCs to test their lipophilicity, then to conduct MTT assays to test their cytotoxicity on a MCF-7 breast cancer cell line. For a bit of background information regarding my project, cisplatin is currently one of the premier anti-cancer agents that utilizes the platinum metal, and you can see the chemical structure of cisplatin here in figure 1. A major disadvantage of cisplatin is that it has severe side effects that essentially limit its use in healthcare. And because of this, there's a need to uncover a new metal-based anti-cancer drug. And this is where ruthenium 2 plus polypyridine complexes come into play, and they could be the potential solution to this. There are 3 major factors that aid the metal ruthenium in being anti-cancer drugs, and those reasons are its ligand exchange kinetics, its affinity for higher oxidation states, and its ability to mimic iron in the body. And on top of these factors, the ligands that are coordinated to the ruthenium metal can also enhance its solubility and cytotoxicity, which is why the bipi and dip ligands were chosen for this project. So the dip ligand is used to primarily enhance cytotoxicity, while bipi ligand is used for improved water solubility. The synthesis of the RPCs involved being refluxed overnight in ethanol, then filtered and conducted a metathesis reaction from PF6 anion to a chloride anion. Then the heteroleptic RPCs were synthesized in a similar manner, just refluxing a 1-to-1 ratio of ethanol and water instead of just ethanol. For the LogP assay, the partition coefficient was determined using the shake flask method, where the solute is placed in a separatory funnel with an equal ratio of octanol water or octanol and buffer. After manually shaking the funnel for 30 minutes, the solution was left idle for 24 hours and then read using a UV vis spectrophotometer. And for the MTT assay, the MCF-7 cells were seeded and grown 24 hours, then treated with the RPCs at various concentrations. The plates were then incubated and left for 96 hours after treatment. Then after this, the MTT was added and then left again to incubate for another 4 hours before removing the cellular supernatants and then adding the DMSO to dissolve the formazan crystals produced by the MTT. And the absorbances were then read at 560 nanometers using a UV vis microplate feeder. Figures 2 and 3 show my synthesized structures, and figure 4 illustrates how the LogP assay was conducted, while figure 5 shows the IC50 results of the MTT experiment. And table 1 sums all that data together as it's compiled to show a better correlation between cytotoxicity and lipophilicity for the dip and bipi ligands. So in conclusion, all RPCs were successfully synthesized and characterized via proton NMR. And based off the results from table 1, Rutris dip has a high log P value, which lets us know that it is very lipophilic. And the Rutris bipi has a negative log P value, resulting in it being very hydrophilic, which means improved water solubility. And again, based off table 1, the more dip ligands coordinated to the ruthenium complex, the higher the cytotoxicity values become. And future experiments will focus on conducting subcellular localization on the RPCs to determine how much of the RPCs get into the cell, and then conducting a compartmentalization assay to see where the RPCs accumulate inside the cell, whether it be in the nucleus, mitochondria, cytosol, or cytoskeleton. Thank you for listening to my talk and allowing me to present in this year's research symposium.